Thank you very much. So I'm Adrian Martin. For those of you who don't know me, I'm from the National Oceanography Centre in Southampton, and it's been a pleasure to be part of the science definition team for exports. So can I just start by saying, particularly for the people on the web, if you've been itching to ask questions about any of the talks, then if you can't wait until the discussion, and particularly if you are watching on the web, feel free to either tweet your question, if you're so minded, or otherwise you can email questions to the email address that you can find on the OCB workshop webpage. So as Mary Jane said, I'm now going to talk about science question 2 on exports. And that's basically what's going on in this red box at the bottom. And so what I'm going to start by doing is just repeating the question that Rue has already showed. And science question 2 is what controls the efficiency of this vertical transfer of organic material once it's left the surface? How rapidly is it attenuated? So this figure is just a blow-up of what you could see in that red box on the previous slide. And what you can see, make sure I point this in the right direction, we have the 5 pathways here. So in the middle, these are the 3 sinking pathways. We have physical transport and vertical migration. So what I'm going to do today is really just to give you a flavor for what's going on here. I can't cover all of this. So what I'm hoping to do is to show you how give you an example of some of the processes that are controlling this attenuation material, hopefully give you an indication of quite how complex these interactions are, and I'll say in advance, this looks a bit sort of messy down here, but it's a lot worse than that in reality. But also to really highlight the fact that regardless of how complex this is, we still actually have a very poor understanding of the first order controls of what's going on within this twilight zone. So I should say, for people who aren't so familiar with the terminology, by the twilight zone, I mean the waters between 100 and 1,000 meters. And that's where most of the organic carbon is actually respired within the ocean. So, science question 2. Within the science plan, this is broken down into 4 sub-questions. So I can leave you to read these, but for the purpose of this talk, I'm going to break those down. So the first thing is the first sub-question essentially covers the whole span. It's really just how this transfer efficiency varies across the 5 pathways. But for the purposes of this talk, what I want to do is to group together some of the other questions. And in particular, the center 2 focus on how the surface community influence what happens deeper down. So even when this material leaves the surface, how is the surface community influencing how rapidly that material is degraded? And then the final sub-question is how does the community within the twilight zone act to attenuate this flux of material sinking through it? So I'm going to start off by talking about surface influence. So in a very simple way, the rate at which material is attenuated within the twilight zone is very simply a function of how quickly it gets across the twilight zone, whether that's physical transport or sinking, and how quickly it is being burnt up, respired, or remineralized en route. And so if we take that simple point of view to start with, then we might, for example, if we're looking at the sinking flux, those first 2-3 pathways of cells, aggregates, or zooplankton fecal pellets, we might think, you know, how do we understand that rate of sinking? And we might think, you know, there are good, you know, physicists have studied this thing for a long time. There are good physical laws explaining how quickly something sinks for a particular depth. For a particular size. So let's look at size versus sinking speed. So let's see how we get on with that. So this is a composite of a number of observations of sinking speed. So on the on the x-axis here, this is size, and this is sinking speed. So you can see that sort of simple physical laws don't get us very far in explaining the variability that we see in the open, excuse me, in the real ocean. So this is admittedly a very simple approach. So one of the things that we don't have in here is what's been termed porosity, the structure of these aggregates. They can be quite convoluted. And the reason this can be important is because it can affect drag. You know, there's a reason that you don't have a roof rack on a sports car. You know. So one of the things that we can consider is the structure, but one of the other things that we consider is density, because clearly, if different parts of an aggregate have different density, this can control the sinking speed as well. And when we further consider that there are, for example, phytoplankton like diatoms and coccolithophores that have minerals associated with them, opal and calcium carbonate, these have higher density, and so this may lead to a greater sinking speed. Similarly, dust into the atmosphere may be increasing the rate at which material sinks. And this is something which has been called ballasting for obvious reasons. You're essentially strapping rocks to these aggregates to help them sink. So even that, it turns out, is rather more complex than we might have first thought. So this is a little complicated, but once again, what we have here is a plot of the size of an aggregate against the sinking speed. And so what I want you to focus on, the two bits that I've highlighted here. So these cluster here, which is of the black triangles, and this cluster here of the open circles, are for aggregates involving the same mineral. This is all using calcium carbonate, but coming from two different organisms. One coming from the coccolithophores and the triangles from foraminifera. And so what this is illustrating is it's not just the density of the biomineral, it's the form it takes. And in particular, one of the interesting things, this was a study using experimental procedure. You essentially put these things in rotating tanks and see how the aggregates form. What they actually found is the case of, if you have these with these foraminiferal tests in them, that actually breaks aggregates up rather than actually helping them form. And so the form of this biomineral can actually have a strong influence, not just on density, but on the rates of aggregation and disaggregation, which are a key control on how rapidly material is attenuated. So the surface community doesn't just affect that process of sinking. I mentioned those two processes of sinking and respiration rate. So it turns out that the surface community can also influence the rate at which material degrades once it's left the surface. This is some nice recent work. So what you're looking at here is effectively some experiments that laced aggregates with this particular type of chemical, polyunsaturated aldehydes, which I will refer to as PUAs from now on, for obvious reasons. And what this is showing is, so the red line is the control. This is your sort of control. This is one-to-one. And you're looking at how the respiration of these aggregates, the amount of bacteria on these aggregates, and the amount of bacterial production on these aggregates changes as you increase the lacing, the dose of these PUAs on these aggregates. So here's the control, one-to-one, and you can see that when you do these first two levels of dosing, then you get an increase, a significant increase in all of these, respiration, cell count, and bacterial production. So these chemicals produced, one of the organisms that produce this, the diatoms, can actually influence how rapidly this material is being respired after it has left the surface ocean. Another way that the surface community can influence the rate of respiration is by exporting itself. So once again, there's quite a bit of detail on this figure, but what I want you to focus on is just this highlight here. This is a tree diagram showing you the degree of similarity between different bacterial communities. And those are communities either within aggregates or free living in the surrounding water. And why I've highlighted this is because it shows you that the most closely related populations are between the population found on these aggregates at 100 meters, so that's material that has just been exported, but the free living population at 20 meters. And in fact, the aggregate attached population is more similar to this than it is to deeper populations, suggesting that a significant fraction of the bacteria attached to these aggregates has actually come from the surface rather than being in situ. So what I've hopefully done there is giving you an idea of the sort of interactions between the pathways and the processes. You can have an organism like diatoms that can affect both respiration and sinking rate, and you can have sinking rate, which involves three of the pathways being involved by a whole host of different organisms and processes in the surface ocean. And that is also a feature of the twilight zone, if not more so. So I'm going to start just with, well, I won't say it's a simple figure because clearly it's not. So this is from a recent paper where they managed to balance the carbon budget within the twilight zone. And so in the top, we have the input flux of carbon. This is in particulate and in DOC. And these fluxes are all in milligrams of carbon per meter squared per day, anybody's interested. And what they then did was to work out, using a simple flow model and the observations that they'd collected, where this material passes between the organisms. So we have detritivores, these are copepods, we have carnivores, suspended POC, attached bacteria, free-living bacteria, okay? And so it's a relatively simple representation of the ecosystem, but just to understand this flow of carbon between them. And what to me is the most interesting thing is that when you look at sort of the largest input, which is the sinking of particular material, the organism which tackles that is not the predominant respirer in this system. There is a tandem process going on. What seems to be the main feature of the system is that the zooplankton are breaking up the aggregates, which is then passing the material, and most of the material is then respired by the bacteria. So you have this tandem effect of zooplankton and bacteria being crucial to the functioning of the twilight zone and the attenuation of the organic material within it. And so just for a few slides now, I just want to focus on what we know about that tandem process between the zooplankton and bacteria. Before going on, though, I just want to mention the fact that, I mean, this is one of the things that potentially exports can look into. This idea that the zooplankton are doing more in terms of breaking up aggregates, slowing down their sinking rate rather than respiring it, has already spawned one, I would have to say, sort of contentious theory, which is the microbial gardening idea. I'm not sure how many people are familiar with this. But the idea behind this is that if you're a zooplankton, then it may well be that fresh living bacteria are actually more nutritious than the aggregate material sinking down, which has probably already seen a couple of owners. That's the nicest way of saying it. And so to them, there may be a benefit in actually breaking up the aggregates, allowing those aggregates to be populated by bacteria. They're now sinking slower, they have a greater surface area, and that then potentially provides them with a larger food source. It's a hypothesis, but nevertheless one which potentially exports can address. So that is a contentious theory. What I don't think is contentious is that we still have a very poor quantitative understanding of this key process of disaggregation, the rate at which these aggregates are falling apart in the twilight zone. So that's why I think that one of the most interesting pieces of work I've seen that comes out recently is one that allows us to get out of sort of the experimental setup and start to look at how disaggregation happens and the rate at which it happens in the real ocean. So this is some work by Nathan Briggs and co-workers, and what he's been doing is using bioargo floats and the sensors on them, not just to measure, so I should say this is depth, and this is essentially the rate of change of the number of particles. Not only is he able to get at how quickly these particles are being lost from the system, so effectively attenuated, but he can also put a number on what fraction of that is actually coming from disaggregation. So we're actually starting to get to the important numbers we need for putting these processes into models. So I've talked about zooplankton breaking things up, how well we understand that breaking up process. Now I want to talk about the bacterial side of things. So I've already mentioned that the surface community can provide attached bacteria to help respire this material as it sinks. The obvious question then is, what sort of impact do they have on the system? So this is some recent work by Anna Belcher. So you've got two sets of 2 bars here. For each set of 2 bars, you're looking at a particular depth range, and the black line shows you how much of the particulate organic carbon was lost within that depth range. So in this case, from 36 to 128 meters, and from here, from 128 to 500 meters, effectively the top and the bottom of the twilight zone. The right-hand side shows how she's managed to account for different processes which may have resulted in that loss. And in particular, one I want to highlight is the fact that she's been able to measure how much of it is due to respiration by the attached bacteria. And what you find is it's actually a small fraction of the total in each case. What seems to be more important is this solubilization, this conversion of the particulate material into the dissolved material. Okay, so we think that potentially the role of maybe the attached bacteria has a lesser influence on the respiration. So then we can look at what's happening in terms of the free living bacteria, the bacteria that natively inhabit these twilight zone depths. Even there are distinctions to be drawn, and in particular, because there are bacteria in these depths, indeed anywhere, which are motile and hence are able to make use of newly emerged organic material much more rapidly. So what I want to show you here is some nice video footage, effectively, showing you how rapidly bacteria can respond to the emergence of fresh new organic material. So on the left-hand side, you're going to see, in 2 examples, it's going to be a dead phytoplankton cell. In the third case, it's going to be a fecal pellet. And the colored contours around it are showing you the concentration of bacteria as they start to move in and then move away. The top right shows you the number of bacteria within a given distance of the cell. So once again, that will allow you to see how they're converging and then dispersing. And then the bottom, this is just the total number of bacteria within a given radius of that organic material, and that's a function of time. So the thing to notice here, this is in minutes. So this shows you how quickly these cells can respond to new material. So let me just start this. So there are 3 here. They're going to go fairly quickly. So if people want, at the end, we can always show those later. In a sec. So you can see, particularly in the top right, this rapid decay in the number of bacteria surrounding the cell. That happens for both of the phytoplankton cells. But when you get to the fecal pellet, you'll see that that population is sustained longer. Okay? So whether that's because the material is more difficult to obtain, because there's more material, we don't know. But clearly there is a strong indication that these bacteria, some of the bacteria, is very able to move to and exploit organic material on a very short timescale. So that, So that has focused on the sort of the zooplankton and bacteria. But that's also highlighted is the importance of understanding the DOC dynamics, the importance of the dissolved fraction. And one of the things I haven't really touched on so far is the pathway of physical transport that can just advect DOC predominantly, but also POC from the surface down into the twilight zone. And I think one of the other many challenges that we face is disentangling that advective pathway from the creation of DOC in situ within the twilight zone. So this is another recent paper by Chris Follett. So this is just take this as an indication of relative abundance. So this is essentially using carbon isotope to work out the age of DOC, different ages, fractions within the twilight zone. And what I want to focus on is the rightmost column in each case is the fresh stuff. Okay, so that is hopefully analogous to the stuff that's recently sunk. And you can see that whether you look at 50 meters, 500 meters, or 2,000 meters, there is a fresh source of dissolved organic carbon at all depths, suggesting that you can't just treat DOC as something that's been advected from the surface. It is being produced in situ, and we need a way of teasing those apart. The whole field of characterizing and separating DOC is a huge one, I know, not something I can go into here, unfortunately. So there are broader controls on, environmental controls on this fate of material in the twilight zone as well. One of the obvious ones is temperature. You know, we all know from physiological point of view that temperature can change physiological rates like respiration being the most obvious one. And so we might expect that temperature has an effect on the rate of attenuation of material. So for people not familiar with this, one of the standard ways, particularly in models and as a way of summarizing observations that people have looked at the attenuation of this sinking flux of material is by fitting curves to it. So one, your flux at a particular depth is essentially a power law of the depth. That's become known as the Martin curve after John Martin, who first came up with it. Or alternatively, people have used an exponential relationship. So these plots show the same data. This is showing you the power law scaling for the Martin curve, and this is the length scale for this exponential. But both are a measure of how quickly this flux gets attenuated with depth. And this is plotted as a function of temperature for the data that this was Chris Marseille was able to collect at the time. And what you see might view as promising that there does seem to be some degree of relationship between temperature and this length scale. But as I know Scott will show you later, there is at least one other study showing you diametrically the opposite relationship. Okay? So even for this basic control of temperature, we still don't even know which way the sign is on the response to temperature. So one thing that Dave and the other people in the SDD wanted me to mention very briefly was other programs springing up that are looking at similar questions to exports. And I'm going to talk briefly about a UK one, but there are also projects. We know there's a project that is going to be running over the next few years in Spain, and we know that colleagues in France, Germany, Japan, and a whole number of other places are also looking at similar questions to exports. This particular project is called COMEX. It addresses very similar questions to export, but the focus is going to be, in this case, in the Southern Ocean. And so what we're going to be doing in that case is looking at broader environmental controls, I think is the way of summarizing it. Around South Georgia, we're going to be using a strong gradient in production to look at sort of how that influences the fate of material. Off Benguela, where we have a strong gradient in oxygen, we'll be looking at the oxygen control. And then the strong difference in temperature between these two locations will allow us to tackle the effect of temperature as well. And just for information, that field work is going to be running in parallel with the projected dates for exports. So that mention of oxygen brings me on to the pathway I haven't really mentioned yet, and that is this, the direct transport of material by migrating the zooplankton. So this is a plot put together by Bianchi quite recently, and this is essentially showing you, so the colors here are showing you the depth to which the zooplankton are migrating. And so that gives you an idea of the depth to which they're going before they're sort of releasing this organic material. And then the contours are showing you the gradient in oxygen between the surface and deeper down. So the greater that gradient, the larger the deficit of oxygen at depth. And so what you see is that you get shallower migration in regions where you have stronger gradients in oxygen. So depletion in oxygen is having a direct influence on how deep these zooplankton are going. What's interesting is this seems to happen even above levels in which you might expect the oxygen levels to actually have a physiological effect. So just to finish, this, I thought I'd sort of go back to the surface. So this is the, I guess, a very familiar image now. So this is from the VIR satellite. This is showing you sort of the variability in phytoplankton that we all take for granted now. Each of these eddies is about 50 or 100 kilometers across, according to where you're looking. But you can see all of this sub-mesoscale structure on this. And as Mary Jane said, we are very familiar with this, and we've realized because of this the importance of understanding the causes and consequences of this sort of spatial variability if we want to understand surface biogeochemistry and export. And so what I wanted to do really was just finish on the fact that, we still don't know what anything looks like at this sort of level of detail in the twilight zone, whether it's bacteria, whether it's zooplankton, whether it's particulate material, or, insert your favorite organism process there. We don't have anywhere near that level of understanding below the surface layer. And so hopefully I've given you a flavor for the interactions, the pathways, in particular, the complexity of what's going on. But aside from that complexity, hopefully I've also got through to you that we still lack fundamental understanding of most of the first order processes going on in the twilight zone. Thank you.